I don't know why. Can you help me to change the slide from, if I say next slide, can you change it? Okay. So can you change to the next slide, please? Yeah, thank you. So dissipate the science advancement taking place through clinical application as well as methods. Next slide. Next slide, please. So the strategy what we are going to follow or we are following for the last 15 years is conducting high quality and value adding workshop, seminars, training like what we are doing here today, which helps pharma industry professionals, academia to enhance their skills and knowledge as well as perform their job more effectively and efficiently. Next slide, please. We also work very closely with universities, colleges, other professional bodies and regulatory bodies, thereby equip the PhD, postgraduates and pharmacy students through training and workshop to understand the modern and advanced dissolution system, equipment as well as software. Next slide, please. Create a value-adding website through which the members of the industry, professional academia can place their issues related to dissolution, method developments, etc., and an expert panel shall offer solution to the issues. We want to create an e-journal, which is still not happened. If somebody can take interest from here and support us, we will welcome that. Next slide. We identify and work closely with young upcoming scientists, chemists, pharmacists from our industry and academia and train them with high quality presentation skills and help them to publish papers and make effective presentation in the national and international forum. So I don't know how many of you are aware here that there is a program for SPDS called DRPI. Have you heard about it? Any PhD students? DRPI means Dissor Research Presentation, started with India in 2020 and became international after 2 years. So today DRPI is a global. So you will go to my website and you will find most of the good international video presentation of the research, original research presentation of the PhD students and masters are placed there. You can view it free of cost. Okay, and you will be, at least SPDS is very privileged that we have brought out to the world many research students who have been doing various innovative methods of dissolution and application using various dissolution methods. And last 4 years, to my knowledge, maybe around 40-50 research scientists might have got the awards from SPDS India as well as international. And we are very much privileged. We have Dr. Pooja with us today. 2 years back, she got the global second prize for the global presentation on dissolution research. Please give a big hand to her. Pooja will be making a presentation of exactly what research she presented and got the second award globally. I mean, competing with UK, Europe, US. And it's not small because there were 35 PhD students from USA participated day before yesterday. So we have these finals coming in September 20th, this weekend. So you're all welcome to join to see the live international competition on research presentation, which has been the best of the best scientists, students present day. Okay. So after seeing her presentation, I need at least 8-10 PhD students and some faculties to motivate the student to make next year, because from Nasik so far we have not got any entry in our DRPI. Generally what happened, all these presentations goes to the big colleges. Big colleges not necessarily be to the best students. That's what my experience. Because there are a lot of good students, innovative students in the periphery of India. India is a big country. So in Nasik also it's an educational hub. So I would like to really thank now Ravi. Ravi is here, no, sitting. Yeah. But for Ravi, we would not have been here. Because Ravi came to know that he is a teacher, visiting faculty. So I said, when he said that in this college, these facilities are there, then I said, why not they ask their interest in conducting a workshop like this? So I'm happy that with his interaction, and then Dr. Swati, Dr. Ashish, Tambesa, all of them took so much interest and organized this. It's not like, as someone said, not another conference. I see a total dedicated teamwork here. When I say this statement, I mean it. I don't see this in all colleges. The faculties can feel proud. Trustees can feel very proud. You will not see so much involvement. The fact that the hard work they have done, the hall is full today. You have 300 plus people sitting and listening today. It's not easy. So yesterday when I met few hours while going to the temple, your principal, Dr. Tampe, as well as Dr. Ashish, amazing people. See, we can find out like the rice is cooked or not. We don't take all the rice, no. We just take one and press it. So I did it yesterday with 2 people, not even 1. Amazing people they are. So I'm very happy. So we would like to definitely add value more in the coming years, not only in the area of dissolution. We have another big society with the heading Rajiv and others, Society for Pharmaceutical Quality Leaders, where the members are only HODs of pharma, QA, QC, R&D, regulatory and manufacturing. So all over India, we will have around 1,000 people, leaders, whom we can bring to the university and give value additions to the students and teachers. So I see this as a very good opportunity here to do that in the coming years. So this is our website, SPDS. So before I leave this floor, I want to put a small video of 6 minutes, which will speak more about SPDS more effectively. But I would like to really express my sincere thanks to all the faculties and trustees of this. And it's really a pleasure that we have the Joint Commissioner of FDA with us today in our, we work very closely with the regulators. To me, the regulator means Dr. Rajiv Raghuvanshi, because I've been associated with him for the last 15 years. So wherever, and Rajiv himself is a very fond of dissolution. He always says, so he has been 2 times our chief guest here. He came as a chief guest in the 2 months back. We had a conference in Indonesia. That Indonesian conference went a very high scale. Our international called Disso Asia, which we did in the FDA of Indonesia, where the chair equal to Dr. Rajiv, Dr. Taruna Ikra, he was the chairperson of the conference. Our ambassador was the chief guest, Indian ambassador. Very good support he gave. And some of the people sitting here, dignitaries who were there with us. We had also similarly 300 plus participants from Indonesia and neighboring countries. So, and that helped basically to make a bridge between India and Indonesia in pharmaceutical area. So we are now working little beyond dissolution, help Indonesian pharmaceutical market, the quality to improve the pharma production of Indonesia through our team members of both SPQL as well as SPDS. So with this, I once again thank the trustees as well as Dr. Tambe, the principal, as well as Ashish and Swati and Madhura, whoever, some of the names, because these people have worked behind. Till today, I have not seen a team from the college going with the letter to all pharma companies. This is amazing to me. It's a learning for me too. So next time when we have a tie-up with MOU with any college, we will put this clause, that you have to go to all companies in person and invite. So that's the reason you will find so many industry people also today who has taken their time to attend to this. I'm sure today's proceedings are going to add high value. Of course, I started dissolution learning from Sotax. Fortunately, they are the leaders in the world. And Sotax is kind enough to bring a USP4 apparatus. For most of you, this may be first time. You may be seeing, you can touch and feel, go and see what are the various cells it has and what are the type of dissolution you can do. If some research students who don't have access to this USP4, don't think that you should stop your project. Just write to us, call, we will give you the instrument Like I remember Pooja can share how many months she worked in this institute, in our lab. She brings the sample, Suhas Evely, our expert, and she makes her data and she makes her project, present the thesis, and today not only she become all India topper, world number 2, and now she is a see all these students, they automatically picked up by foreign universities. You just apply, say that you want the SPDS award, because SPDS judges are global, not Indian. No, they are placed across, and they're all from AAPS or FIP, big associations. So that's a very good visibility when you participate through SPDS in our DRPI program for academic research. So please utilize it. I would love to have next year 3-4 students from this college. So I will ask Dr. Tambe sir, how many students from your target is at least minimum 3 students should participate. Winning is not important. Participation is more important. Everyone cannot win, but when you participate, you learn a lot. Okay. So with this, I stop and I request to play the video of SPDS. So it's a short video. It will take care of the last 15 years of our journey. Yeah. Can you?
Defined by complexity and rapid change in healthcare, the pharmaceutical industry depends on cutting-edge technologies, robust regulatory frameworks, and the expertise of visionary professionals to drive meaningful impact. Welcome to the Society for Pharmaceutical Dissolution Science, SPDS, a global platform where scientific innovation converges with regulatory excellence to advance pharmaceutical quality worldwide. Established in 2012, SPDS was founded by forward-thinking leaders from both academia and industry, united by a shared commitment to elevate the science and applications of dissolution. Spearheaded by our esteemed founder and past president, Mr. Vijay Shivsagar, a veteran in quality, regulatory affairs, and analytical sciences with over 4 decades of experience, SPDS has grown into a beacon of excellence. Joined in this mission is Professor Padma Devarajan, our current president and a celebrated academician, scientist, and innovator, and Dr. L. Ramaswamy, the founder and Secretary General, whose lifelong dedication to pharmaceutical quality continues to inspire the community. SPDS continues to grow through powerful global partnerships. Sotax, a world leader in dissolution testing, has been a strong supporter since our inception. So much so, on our foundation day in Mumbai, Mr. Rolf Benz of Sotax reaffirmed this alliance with a personal endorsement.
We at Sotax are very happy to be associated in all these years in the scientific programs of SPDS.
ACG, the world's largest integrated supplier and service provider to solid dosage manufacturers, proudly stands as a key supporter of SPDS. Mr. Ajit Singh, chairman of the ACG Group, is the chief patron of SPDS.
The great efforts made by the SPDS and the FDA of Indonesia that we are able to now take part in a very significant event.
We are proud to state that SPDS registered office is based out of SciTech Center, which is the R&D affiliate of ACG. Under the leadership of Professor Padma Devarajan and Professor Munira Momin, Principal of BNCP and Scientific Chair of SPDS, we are setting new standards in scientific and regulatory collaboration. Today, SPDS is a global force. We expanded to the USA in 2019 under the guidance of Dr. Vinod P. Shah, former US FDA scientist and respected consultant. Our flagship annual events, DISSO India, DISSO US, DISSO Asia, bring together 14,000 plus delegates from regulatory bodies, scientific institutions, and research organizations. To empower the next generation, Dissolution Research Presentations International gives young researchers and students a global platform to present, learn, and lead, thus gaining visibility and receiving valuable feedback from leading experts. Each year, SPDS honors outstanding contributors in the field of pharmaceutical sciences through the SPDS Award of Appreciation. This prestigious recognition is presented to renowned educators and scientists from academia and industry, whose work have significantly advanced drug quality and dissolution science. These individuals embody the mission of SPDS to ensure safer and more effective medicines for patients around the globe. Knowledge dissemination is central to our mission. Through collaborations with top universities, regulatory authorities, industry leaders, and professional bodies, we conduct high-impact international scientific conferences, training sessions, interactive workshops, and expert forums. Our educational initiatives span the latest in dissolution science, regulatory practices, and state-of-the-art technologies, including automation and analytical advancements. Our flagship publication, Desk Book on Dissolution Science and Applications, is a trusted global resource. SPDS is proud to be at the forefront of a global movement that is redefining the role of dissolution science in drug development and quality assurance. We invite professionals from across industry, academia, and regulatory sectors to join us in shaping a future where safe, effective, and high-quality medicines are the global standard. Step into a world where science meets strategy and innovation meets impact. Join SPDS and be part of a community committed to transforming pharmaceutical science for the better. SPDS, advancing science, ensuring quality, empowering the future. We look forward to welcoming you to our growing global community.
Thank you, Ramaswamy sir, for the brief introduction of SPDS. We are glad to hear about the imminent work done by SPDS. Your vision and mission will surely help the science world to achieve the goals. May I now take the opportunity to respectfully invite Dr. Padma Devarajan ma'am, our distinguished president of the session, to kindly come forward to the dais and deliver the presidential address. We look forward to your words of wisdom, ma'am.
So we are giving that 2 edition, one goes to the college, a complimentary copy from our end.
A very good morning to one and all. At the outset, let me thank, on behalf of SPDS, the MGV College and the Hire Knowledge, not audible? Yeah, okay. So it is indeed an honor for SPDS to be part of this joint collaborative international conference. We have done many conferences with other institutions, but the kind of numbers we see here and the enthusiasm that we saw before the conference certainly has made us very excited about today's entire program. I would also like to greet the FDA Joint Commissioner, all the management and leaders of the foundation that is running these institutions, all the leaders from academia, all the speakers, including the speakers from abroad who have very kindly consented to be here online or in person like Pooja. And also I would like to thank all the faculty of MGV for their very valuable support and cooperation so that we could reach this point today with great success. And we also know that a great beginning is 50% done. So we are left with only 50% of success left. And I'm sure the rest of the day is going to take us there. We've seen a wonderful video on SPDS, which has said a lot of things. But what I would like to focus on is just one or two things, because I can see there are so many students here and teachers here. SPDS is a society that is a collaboration between industry and academia. And that's how you saw the founder, Dr. Ramaswamy, here with about 40 years experience in the industry. And I stand here as an academician with about 40 years of teaching experience, not only teaching, but teaching, research, innovation, and a vast experience which is going to come through you through the entire SPDS fraternity. So what is it that you are going to benefit from SPDS? SPDS runs conferences like this, which are local conferences where you benefit through knowledge exchange, through a platform where you can interact. So I would like to tell you that during lunch, during the tea break, do not hesitate to come and talk to any one of us. We find that students want to talk to the speakers, want to talk to industry leaders, to the academicians, but there is a lot of hesitation. This is a platform where you should use this to get over that fear and come and chat with us, because that's what we are here for. We want to understand what you want, what more we can give. We have a conference annually called DISO India, which is a conference where we have many more international speakers and speakers from India in a focused part of most of the time a Tier 1 city. That gives you an opportunity to come to that city. We also have most often poster presentations there where you could present your posters. A special focus is what Dr. Ramaswamy just told you about, DISO research presentations, India, which has gone international. In fact, the finals, that is DRP International, is going to be held on 20 and 21st of this month, and it's entirely online. So we will share the link with you, and we would love to see you participating. It's one of the best opportunities you can get to understand. Coming to the one word dissolution that this society focuses on. Now, is dissolution just a small ambit of formulation development? No, it's a very specialized ambit of formulation development. And it doesn't start when you put your tablet into the USB or IP apparatus. It starts when you start thinking about a formulation. It starts when you want to develop a formulation. So dissolution is a concept that has to begin at the beginning of a formulation, so that when you're done with your formulation, you don't have a dissolution challenge. So dissolution is not something small. It's something very critical, something that's very important to ensure that the tablet I take works. So it's not something that is just to be thought of as a test, a routine test, a quality control test. A very important aspect of dissolution today is the immense innovation. I heard the anchor talk about quoting Abdul Kalam, our former president, where he said innovation is very important. And I was also given the impression that this college believes in innovation. And I think one area of innovation all of you could get into, particularly those into pharmaceutical formulation development, is innovations in dissolution. To give you just one or two examples, you can look at different media for dissolution, where you have biorelevant media. You have today, beyond biorelevant, clinically relevant media. And these kind of tests would tell you in vitro what could be probable success in vivo. It may not be 100% success, but it could be an 80% success, beyond which you need very minimal in vivo studies to achieve the final formulation. There are very many dosage forms, which Dr. Pawar mentioned in his first speech, and he talked of so many novel formulations. We have a whole horde of nano formulations coming into the market. We have injectables coming in, long-acting injectables. So other than oral, where dissolution testing is standardized with 7 apparatus, transdermal with 2 apparatus, for all these different kinds of complex formulations, today you don't have proper dissolution testing methodology in the pharmacopoeia. That's one huge area where you could focus and become very innovative. And to become innovative there, you don't really need to have high-tech instrumentation. You can do it very, very simply. And when I give my next lecture on nano formulations, I will tell you how we have done that in a very simple way by using simple USB apparatus too. So when we talk of dissolution, do not think it's a small word. Do not think it's a small ambit of formulation development, but understand that it is a crucial, critical part of formulation development, where you could start looking at dissolution from selecting your API. Which polymorph am I going to select? Am I going to select a crystalline or an amorphous form? Or am I going to look at particle size right at the beginning? Or only when I don't see the dissolution happening, am I going to look at particle size? So if you look at dissolution through and through as something very important, you will come up with many more ideas. And to get more ideas, all you need to do is read. So to all students here, I would also like to tell you and inspire you that innovation happens not only to those, to the others. Innovation is something that you can do in your lab, provided you think first, I can do it. 2, when you are doing your research, you look differently at your research. So innovators, I would like to quote, don't see different things, but they see the same things differently. So when you are performing an experiment, if you see a gel where you don't expect it, you have an innovation in hand. If you see a release that is supposed to be fast but is very, very slow for some reason, you have an innovation in hand. Some of those could turn out to be just problems, but many of those could turn out to be your innovations. So I would inspire you henceforth, when you are doing any research, is to look differently, look thoroughly, look completely. So with this, I welcome all of you to this wonderful program that we've organized today with the Mahatma Gandhi Vidya Mandir's College of Pharmacy. We are very honored and privileged to be here. Thank you so much.
Thank you very much, ma'am, for your time and lighting address. Your encouragement and wisdom mean a great deal for all of us. Now, we are truly honored to have with us today our chief guest, Dr. Yogita Tai Hirya. Though she will not be addressing the gathering formally, she has conveyed her warm wishes and wholehearted support for today's conference. Dr. Hire extends her heartfelt congratulations to all the participants, organizers, and encourages us to continue striving for excellence in the field of education and service. Now, first and foremost, a sincere thank to our chief guest, Dr. Yogita Tai Hire, for her gracious presence and encouraging support. A special thank you to the guest of honor and patron and office bearer whose guidance has been instrumental. We also express our deep appreciation to the distinguished dignitaries on and off the dais, industry experts, FDA officials, and all the academicians who joined us today. Your valuable presence has truly enriched this occasion. With this, I officially conclude the inauguration ceremony and we will shortly be starting with the technical session.
Until we start our technical sessions, let's have a look at a glimpse of Mahatma Gandhi Vidya Mandir's Pharmacy College.
Welcome to Mahatma Gandhi Vidya Mandir's Pharmacy College, an autonomous institution of excellence where tradition meets innovation and learning transforms into leadership. Established in 1993 and affiliated with Savitribai Phule Pune University, our college has over 30 years of academic excellence. We are NAC A+ accredited with a CGPA of 3.28, NBA accredited, ISO certified, and approved by the PCI. We offer courses like D-Pharm, B-Pharm, M-Pharm with 5 specializations, Pharm D, and PhD in Pharmaceutical Sciences. Our structured curriculum as per NEP 2020, backed by modern facilities, academic expertise and pedagogy tools, prepare students for every stage of their professional journey. Centrally located, accessible campus, highly qualified and dedicated experienced faculty, technologically advanced laboratories, over 300 plus research publications, 25 plus patents, 20 plus authored books, and 48 plus MOUs with industries and institutions, and a vibrant student life supported by students' clubs, co-curricular and extracurricular activities. We're committed to advancing pharmacy research. Our placement cell plays a vital role in shaping students' careers. We partner with leading pharmaceutical companies, hospitals, and research organizations to provide campus recruitment career counseling. With strong alumni involvement, students benefit from mentorship and professional networking. Our alumni are shining worldwide, from the USA, UK, Australia, and many more. They're leading in pharma research, manufacturing, clinical trials, entrepreneurship, and innovation. Their success is our pride. Admissions are now open. Your future in pharmacy begins here. So for the details, visit us at Hire Knowledge Campus, Panchavati, Nasik. Explore our website, mgvnskpharmacy.kbhgroup.in.
Thank you. Let's begin with the conference. We have 5 technical sessions and a special highlight. We also have a live demonstration of USP Type 4 apparatus by the SOTAX India team, giving delegates hands-on exposure to advanced equipment. The very first speaker is Mr. Vijay Shrisagar. I request Laiba Sayyed to introduce our speaker.
We are truly honored to welcome Mr. Vijay Shrisagar sir, an accomplished quality, regulatory and analytical profession with more than 38 years of rich experience in the pharmaceutical industry. He has held senior leadership positions at renowned companies such as Ranbaxy, Sun Pharma, Lupin, and many more, where he last served as an executive vice president and later as an advisor for quality and regulatory affairs. Mr. Shrisagar sir has successfully led numerous international regulatory inspections by agencies including US FDA, MHRA, and many more. He is also the founder president of the Society of Pharmaceutical Dissolution Science, which has now grown into global platform and has been honored with multiple prestigious awards, including the Outstanding Analyst Award by the IDMA and the Leadership Award by the SPDS. As the founder of Track Pharma Consultancy, he continues to guide pharma and API companies worldwide in regulatory compliance, CGMP training, and strategic consulting. With his deep expertise and inspiring leadership, Mr. Shisagar sir has made an indelible mark on the global platform landscape. Please join me in extending a warm welcome to Mr. Vijay Shisagar sir. With this, I sincerely request Dr. Tambi sir to felicitate our honorable guest, Mr. Vijay Shisagar sir. I now request our honorable guest to kindly commence the session and enlighten our delegates.
Namaste, good morning. So I was to say good morning to you, but now I have to say good afternoon after that because we are 7 minutes after that. Fine, okay, no problem. Important is to deliver and understand. So I'm going to talk to you about compliance challenges, very specific talk. So to begin with, these are my thoughts, of course, and cannot be construed as organizer's opinion or the regulatory opinion. And I do have some rights to that. So what I'm going to show you about some case studies and learning through those case studies. And finally, important point for me is to send you the messages, what you can do at your end to correct. Theory, we have learned a lot from all the research that is going on, all the papers which are published, and all the textbooks that you have. But I'm a hardcore industry person, and I believe in passing on the acts that you can do, the tips that I want to give it to you, so that you are influential and effective person on the shop floor. First of all, why dissolution is important? You heard that we started this particular SPDS in 2012. And initially we had some concern. You just heard from Dr. Ramaswamy, our key person due to which SPDS came into existence. But then at the backbone, I am from the industry. I knew that it is way back in 2012. When the product goes for bioequivalence studies, our confidence level was just around 50%. R&D person used to think, I don't know whether it will pass, it will fail. So we thought, can we help industry to gain that confidence from 50% and raise it to somewhere around 80-90%? And we are happy that we thought all the efforts and the contributions of veterans that we have joined and we have got access to, like Dr. Vinod Shah and others, fairly a good deal has been through. And today we have a much, much higher compliance rate. Thanks to the efforts of all of you, and we are happy to have our good share in that. So it's an in vitro test. First of all, we cannot do these studies for each and every batch, right? Otherwise, we cannot sell anything. So we want to understand that when I take a particular tablet, what happens inside the body? And it should mimic whatever in vitro test I do, it should mimic what happens inside the body. And that will give me the confidence. Vital quality control tool contributes towards quality, safety, and efficacy. How? I think subsequent slides will tell you about that. And ensures batch to batch consistency. Remember, today quality is not compliance. I repeat, quality is not compliance. Quality is consistency. You must get the same result with every batch. Patient must be happy with every batch that you sell into the market. And that's what we are trying to achieve. Why compliance is important? Compliance is important because customers has to be satisfied. Who is my customer? Not only the patient, including the distributor, the seller, the doctor, the prescriber, everybody is my patient, and all of them should be happy with my product. India is a pharmacy of the world. And can it remain pharmacy of the world? There are a lot of threats which are coming in. So we have to see customer satisfaction from all angles. That includes quality and cost too. It's not only important for growth, but its survival itself is at stake. Mind well, survival. It is not only growth. You can grow only if you survive. Today, if you are having a compromise on compliance, your survival is at stake. You must have heard just a day before one of the companies got warning letter in India, one of the leading companies. The same cycle happened a couple of weeks before. I don't know what is going to happen in future. We heard Mr. Donald Trump, who is after all the Indian market into the US, and I don't know whether it is going to get restricted only to the technical part or it is going to get into the economic part also. But anyway, all said and done, as long as we are strong, nobody can hurt us. With regulatory observations, you probably be LTF instead of FTF. What does LTF stand for? Last to file instead of first to file. Today, we are all generic players. We basically copy the innovator and then sell it to the market. So our existence depends how fast we are able to file. And if we are not able to do that, we are gone. And lastly, if you have a compromise on compliance, if there is a warning letter, some regulatory action, it is known to the entire world. What happens with US FDA is known to WHO. What happens with WHO is known to ADV. Every authority comes to know about it. Your global reputation is harmed. So from that point of view, it is very important. Now, I was in dilemma how and what I should talk to you. Because the message must go rightly. It should not be less, it should not be more. So I want to understand, I want people from the industry to raise their hands. So I want to understand how many people are from the industry so that I can accordingly concentrate on those relevant elements. Please raise the hands who are from the industry. Oh, thank you. So I think that is less than 5%. So accordingly, I will frame my discussion so that I am able to deliver the points of interest to both of you, that is the students as well as from this industry. And coming to the students, there are from different colleges. I want to ask you, the students who are present from different colleges, how many of those colleges do not have a dissolution apparatus in your lab? Please raise your hands and don't feel shy about it. I work for some colleges in Mumbai, and one of the colleges for which I work, they also didn't have the dissolution apparatus. So I told them to procure one, then I connected them to Dr. Ramaswamy, and then who gave the very competent quotation to them. So please tell me how many of the colleges who are presently represented here do not have dissolution apparatus in their lab. Please raise your hands. Do I presume that all of you have dissolution apparatus? I saw a short presentation on your college. I did not see the dissolution apparatus there. I was very keen to see that. But anyway, that may not be featuring in your film, but you may be having it. So if you have the dissolution apparatus, you must know how to use it. And that's what we are going to concentrate. And let's see what I have got to tell you about this. What is dissolution? Is it a science which is recent science? It's a science nearly 100-year-old. Can we see the journey? It all started way back in 1897. The purpose of showing this slide is to tell you how the dissolution size has evolved. So, first these two scientists, Noyes and Witness, published a paper on rate of dissolution of solid substances in their own solution. Then 1930, some experiments began. 1934, DT was introduced in the Swiss Pharmacopoeia. 1950, it became official in US. Then 1960, USP began experimenting with a variety of basket and stirring devices. And then 1960, these two scientists utilized a beaker blade stirrer and found significant differences. Aspirin was the starting point. Yeah? Aspirin is acetyl salicylic acid. Actually, if it gets released into the gastric fluid, then you will get a lot of irritation. It has to get released at the right side. And that was the starting point that why some of the products are good and some of the products are bad. All of them are containing, say, 500 milligram or 250 milligram of aspirin, but still some products are good, some are bad. So that was the reason which was found here. Now, major event, 1970, USP 18 introduced official dissolution test. And then 1990, we have the development of paddle method, rotating cylinder, and reciprocating distribution in place. When I also worked for Indian Pharmacopoeia on the expert committee, and we were thinking that we should globalize our all chapters. First, we were calling paddle apparatus as apparatus 1, and we were calling basket as number 2, but worldwide it was called other way. So we said, let's harmonize. So now the latest IP which you must be saying is fairly harmonized from this angle. And 1990, you must see the development. It became USP4 method in 1990 because it was found to be very, very useful. And you are good that Sotax has demonstrated apparatus here, and they are the most reputed manufacturers of this apparatus. Make use of that, try to understand, because this is going to be helping you a lot in future. And the reasons I'm going to tell you in my subsequent slides. So let's take a quick review. Where are we right now? I'm concentrating on the IP chapter 2.5.2. You have 1, 2, you have 3 and apparatus 4. We don't have 5, 6, 7 which are there in the other pharmacopoeias, but currently we are concentrating on these 4. And even if you are able to understand these 3 and 4, I think a lot is done there. Now we have convention release dosage form, immediate release, then there is S1, S2, S3 provision, so on and so forth. And for prolonged release, we have a provision of L1, L2, and L3. Sometimes you hear why these provisions are made. Friends, the reason is that there is a lot of inherent variability, and that's the reason why we have this issue. Now look at this recent stuff. Always I try to gather the current data. I think this is nearly, I think, the 5th time that we are talking to college and academia on behalf of SPDS. We had similar conferences first to begin with Indore, then we had Mumbai, then Pune, then Haryana, and now we are here at Nashik. So you can understand that June 25, May 25, April 25, you see the results of analysis by our state laboratories and the central laboratories, right? So in June 25, let me explain you one line. Based on that, you can read the remaining lines. In June 25, 55 samples were tested by CDSCO, Central Authority. Out of that, 8 were found to be non-compliant from the dissolution point of view. Similarly, the state authorities conducted the analysis of 130 samples in the month of June 25, and then we had nearly 20 non-compliances. So this is May 25, April 25, and whenever I gather this data, every time I speak, I find the results are almost same. And we have about 25% non-compliance if you take the grand average of all. What does it speak? It speaks about many things. Either we don't know how to do the dissolution test, or our dissolution tests are done for the product which is not consistent in quality. So once it fails, next time it passes. What are the reasons for that? And remember, the molecules which are involved there are prominent molecules I have listed down: albendazole, sartans, paracetamol, bimeclamide, so on and so forth. And you can see from that these molecules are mostly belonging to BCS class 2 and BCS class 4. BCS, for the benefit of the students, you must know what is BCS. Biopharmaceutical qualification. We say highly soluble, highly penetrable, then low soluble, highly penetrable, so on and so categories are there. So coming to class 2 and class 4. Class 4 is least soluble and permeability also least. That comes in class 4. Very, very challenging type of molecules. So you can see that around 90% of the small manufacturers are involved in this, but 10% are also bigger companies. So the future lies that you as students, you should prepare this. So when you go to the industry, you are able to do justice to this particular topic. And why class 2, class 4 are important, I think I may like to take you to the next one. Look at this circular. I read this circular just 2 days back. What does it say? That for class 2 and class 4 drugs, the manufacturer must submit the proof of bioequivalence. So this is a very, very major decision which had just come 2 days back. And I think all of you must take cognizance of that. Now, this was the circular of 2017. But it looks like from this particular announcement that it was not implemented. So now the DCGI has to remind the industries again and the state authorities again that please see that you are compliant from this perspective. It was just 2 days back. And it talks specifically about BCS class 2 and class 4. And you have seen in my previous slide that most of those molecules which are affected are represented in these two classes, right? So here is a big, big challenge. And if we want to maintain our prominence, we'll have to take the cognizance of this. Look at this warning letter, very recently issued. I'm showing about the last. I have at least 5 to 6 warning letters to show you, but I'm showing only one due to paucity of the time. One of the leading Indian companies, and what does the warning letter say? That you failed to adequately investigate and resolve dissolution failures, particularly concerning so and so. We are not interested in the name of the company. We are also not going to talk about the name of the products. But it's the ER capsules, and company had attributed this to API particle size. They also said because of process changes, these things have happened. But when regulator scrutinized that, they found that this is not true. Company is not diagnosing the right problem. The root cause is not identified. And that's the reason company got. And there were repeat violations. And there were many more cases of warning later because of dissolution non-compliance. So we are talking about a very important topic. So what we can do? What is the CAPA for us? So let's talk about that. We have to talk about the current nature of quality, which is compliance plus consistency. It's not only compliance, consistency. So that's the reason 1980, when the product failed, it was a forced dissolution testing. I was a chemist in 1980, working for a company which is among top 3 today. What I used to happen, that R&D guy used to send me the sample, and I used to test and say, sorry, it's not showing the right dissolution. Then the R&D guy says, look, what is the SLS percentage you use? I use 0.25. Do one thing, make it 0.5%. Do the test. Sir, it is still not passing. Okay, then do one thing, make it 1%. And then I used to say, oh, it's passing. So it was a forced dissolution with no relevance to what happens inside our body. So we have to get rid of that, and this is the case. And remember, testing 6 or 12 tablets out of a batch size of 5 lakh, 10 lakh, can you really predict what is going to happen with the entire batch? No way. So what is most important is the confidence, and confidence we are trying to get through. So we have to take the cognizance of all these. We have to see how it disintegrates, how it disperses, how it dissolves, how it gets diffused, and how you can do it when you are first time studying the product, especially in the R&D level. You must see right from the start till end. So there are cameras which are available today, automation is available. You can see how it integrates, how it diffuses, and a lot of things will come to you if you study all these aspects carefully. There are procedural changes which are required. I am going to tell you about what is the current understanding, what are the latest, some findings which are really mind-blowing. We just come to know that deaeration has to be validated. Some products may need, some products may not need. Currently, you are understanding that deaeration is must for every product. But there are a lot of cases, I cannot dwell on that due to paucity of the time. Ensure proper centering of the tablets. I have some case study from USP where they are talking about proper centering of the tablets. When you put the tablet into the apparatus, it must get into the center. Now, there is no way you can do it. Currently, when you drop it from the top, it can be off-centric slightly. But there was a case which we took a USP and USP said you have to ensure. How? They also don't know, but we have to ensure it. Explore the use of peak vessel. We talked about peak vessel. This is not an official vessel, but many times this will help you at the R&D level to understand the nature of the product. Implement QBD, quality by design. It's a science by itself. Only with QBD you can ensure consistency. Otherwise, it's only compliance. Method changes and guidances, and we can become trend setters and not just followers. Lot of threats are there. India is being seen as a very, very competing nation today. Across the world, we are going to get into the top 4, top 3 sectors. So there will be a lot of people who will be watching us and they will try to create a lot of problems. But nevertheless, we have to maintain our dominance. Oh, this is... Now let us look at what has changed significantly. In dissolution target, it was 100%. No, we already discussed that it has to be discriminated. It can be 70%. Innovator concept almost was non-existent, but today you have to ensure that your product is comparable to the innovator. That's most important. IV-IVC, most prominent. You must ensure the IV-IVC. We talked about the latest circular. Test method. Pharmacopoeial methods are not necessarily the best methods. You need to have your own methods which are discriminatory. Please remember that. Dissolution limits. Pharmacopoeial limit may be 70%. So your limit cannot be 70%. Whatever studies you have done for bioequivalence, if that has given you 85% dissolution, then your limit has to be 85%. Dissolution points, it cannot be just one point. You may have to use multiple points, and that should be the part of your normal process. Dissolution range, based on actual dissolution with little or no correlation to in vitro performance. From that, we have to move. The limits have to be such that required IV-IVC is achieved. Method principles, whether you use UV or HPLC, you should be fully justified. If there is no interference, go ahead with UV method. If there is an interference, you have to use HPLC. Method development, we talked about QBD. ICH Q14 is the latest guideline which has come. It talks about the implementation of quality by design for analytical area. Now there are some provisions. I want to tell you that be careful about that. Don't use them just like that. It allows that where dissolution test is specified in the individual monograph, DT is not required. That's official provision. But my recommendation to you, please validate this. And it is good to study that. As per ICH Q6, DT can be substituted for dissolution in dissolution more than 80% in 15 minutes. Highly dissolving. But again, word of caution. Substitution is also allowed when the DT is more discriminating than dissolution. That's the pharmacopoeia provision. But my suggestion, verify. Don't go by pharmacopoeia provision. You can land into problems. Now, let's go through some of the regulatory queries which will tell you where we are right now. Okay? High dissolution rate was obtained, and company said it is because of high weight of the tablets, and say regulators said it's not just EPA. When you do the dissolution test, you have to select the tablets randomly. You cannot weigh them and test it. If you weigh and choose the tablets which are good for dissolution point of view, sorry, you're not doing the right testing. No separate field alert for dissolution. Whenever you have a problem in dissolution which affects the clinical efficacy of the product, you need to raise the field alert. Even for our domestic products, it's necessary now. Sufficient quantity of the control sample is not kept. This is a common problem because you have S1, S2, S3, then L1, L2, L3. You may need 6 plus 6. Plus 12 plus 24 tablets, so you should have adequate number of tablets. Deaeration, as I mentioned to you, it has to be done only if it is required. If it is not required, deaeration is not required. Are you able to hear that row? And as I think this is told you that SPDS, this program is just the beginning. So we like to for a long time, whenever you have any issues, any problems, you are most welcome to contact us. So that's how we really want to collaborate. And our sole effort is that whatever you work leads to a meaningful development which can be commercialized. Validation did not cover accuracy of dissolution method. That's a very challenging test. Now, how do we do the accuracy? You have to make the placebo, you have to add active ingredient to that, make a solution. You know that this is the amount of active I have there, then you charge it in the vessel, then you try to get it back and say, oh, accuracy is there. Then low dissolution rates are attributed to detachment of the basket from the shaft. Analysis is not done any note of it during testing. So whenever you do the testing, please understand you have to record everything in a contemporaneous manner, not subsequently. The moment you do anything, that has to be documented. And that's the hallmark of compliance today. Whenever you do that maintenance activity, you have to do the calibration again. But many times you don't do it. at college level, you may be maintaining your dissolution apparatus. So do not use it unless you calibrate it again. Water, whatever is used, whether in the bath or the apparatus, has to be clear. So if the bath is showing turbidity, that means it is having some fungal contamination, and that can lead to misleading results. Then, as per the manufacturer, batch failed at 3 months accelerated stability condition due to electric failure for 10 minutes, simulation studies were not done, and root cause was not conclusive. Stability testing in your stability studies and dissolution testing in stability studies, these two need to be very, very carefully evaluated. We have a big problem in the industry of not doing the stability testing for dissolution and many other things in a timely manner. And dissolution stability stands out amongst the top non-compliance. People do assay, RS and all those stuff, but for some reasons, dissolution is given the last priority. And this is what the regulators have found. And the reasons which were given by the manufacturers were not justified. Please introduce intrinsic dissolution test for the API. Nowadays, dissolution does not get restricted only to the formulation. Please understand what I am saying. I am talking about the dissolution of the API, active pharmaceutical ingredient. There are 2 chapters which have been introduced now in the official pharmacopoeia. You have apparent dissolution, you have intrinsic dissolution, and this is the apparatus which is widely used to carry out this particular test. So there is USP 1087 which talks about intrinsic dissolution, then PHER talks about apparent dissolution, and more than 90% of the molecules are found insoluble today. That's why this is going to be a major driving factor. Validate the use of UV or HPLC as we talked about. If there is an interference, do not use UV, use HPLC. And wherever there are any changes in materials, please revalidate your method, because those materials could be creating interference in your estimation. And method validation nowadays, even our new Schedule M is giving a lot of stress on method validation. Earlier it wasn't so. But now the new Schedule M, which is at par with WHO, which I hope you must have read it at least once. This is going to be an important reference for us, and that talks a lot about method validation. Whenever you have a BE failure, for the industry people, have a procedure of investigation. Currently, what I've seen, when a BE is failed, it's only restricted to the R&D. But I'm suggesting that it should be a wide effort and everybody must add value to that. And that's where the concept of monitoring became important. And when I was a corporate quality head, my job was not only to evaluate, but I also involved in the monitoring of the bioequivalence studies. Dissolve percentage and undissolved percentage. You estimate only percentage dissolved, right? But you should also, as a matter of study at least the R&D stages, estimate what is percentage undissolved. So make the mass balance there clearly. Dissolution training needs to be demonstrating. What does it mean? Man, just don't call your students or the trainees and talk to them about dissolution methodology and chapter. Please see how they are actually doing on the shop floor. Stand beside them. Then you will find they're not reading the tablet properly, they are not doing the deaeration properly, they are not ensuring the vacuum properly. So many things will come to the fore if you have demonstrative training. Our old concept of me teaching you, telling you, and then you answering my question papers has gone for a toss. It is meaningless actually. So I must see how you do actually in dissolution in your lab. And if I see that yes, my talk has really resulted into a lot of care being taken at your end, then I will say my training was successful. Otherwise, no. This is my own thought. This is you can say this is my patent, which I have been promoting through that don't challenge only passing results. And don't challenge only failing results. Challenge both. So far, when you get the OS result, you challenge it. But as long as it is passing, you don't bother. And when you get the OS, then you say, oh, chemist did the mistake, and due to which actually product was good, but it failed because of mistake done by the chemist. Now, my simple question is that, can you not go by the reverse logic that the product is bad actually, but because the mistake done in the analysis, it has come all right. So I have been talking a lot about this in front of US regulator, EU regulators, everybody I talked to, they all liked it. Now I'm going to take it to a larger platform. So at least I think I speak at least 25 times in a year in various conferences. But every conference I make it a point to put one slide on this because I want to drive home this important message. So these are the useful references which I got through. Finally, big thank you. I must thank Mahatma Gandhi Vidya Mandir Pharmacy College for taking this initiative and giving us an opportunity to be with you. Really grateful. Thank you so much, Dr. Tambe, Dr. Pawar, Dr. Swati, and so many. And thank you for your keen interest. And hope to see you again. Bye-bye. Thank you.
We are deeply grateful for the wisdom and clarity you brought to your talk. Thank you for your valuable time, expertise, and dedication. We deeply appreciate your meaningful contribution to the conference and look forward to the future opportunities to learn from you. Now, I would like to invite Dr. Pramod Sir, Principal of ASS IIPT Nasik, and Dr. Vijay Srisagar Sir on the dais. I request Dr. Katkadi Sir to felicitate Dr. Vijay Srisagar Sir. Thank you, sir. It is my privilege to introduce Dr. Padma Devrajan Ma'am, a distinguished
There is a slight change in the plan. We are now having a lunch break. I request faculty members and industry experts to have their lunch first. And until then, I request all the students to look after the SOTAX team is going to present a live demonstration of type 4 USP apparatus, dissolution apparatus. Kindly have a look at that. And after that, we are going to display some videos of vibrant campus of the vibrant Hire Knowledge campus. Also have a look at our KBH Dental College, Hotel Management College, and LVH Art Science and Pharmacy College too. We will reconvene at the given time. I request until then all the faculty members and industry experts to have their lunch. And students, please visit or have a look at the live demonstration by SOTAX team of type 4 USP apparatus. Thank you. We will resume with our sessions on 2 p.m. Until then, have your lunch enjoy the lunch break. We are also having live demonstration, enjoy that. And students also can go for lunch break.
This is the water bath where the temperature is get maintained. Then inside bath, this is the heat. Under this is the heating jacket.
Yeah.
ऑडिटोरियमचा पासवर्ड काय आहे?
I think sure. It's okay. Thank you.
welcome Dr. Samir Haduchi sir, an eminent chemist and entrepreneur who has pioneered advancement in dissolution testing. Beginning his career with Sandoz Agro and Novartis, he later founded SPS Pharma Services, the world's first CRO dedicated to dissolution and release testing. Today, SPS operates globally as part of the Sotax Group, where Dr. Haduchi sir leads the pharma services segment across Europe, America, and Asia. A recognized global authority in vitro dissolution, IVIVC, and laboratory automation. He is frequently invited to the international conferences and also advises scientific societies and health authorities. Dr. Samir Haduchi sir is joining us virtually from France due to his busy schedule to deliver the insightful session. Let's warmly welcome Dr. Samir Haduchi sir. We are felicitating sir by the online mode. We welcome you, sir. I now request our honorable guests to kindly commence the session and enlighten our delegates.
Yes, hello. Yeah, hello everyone. I see some familiar faces in the audience. Good afternoon. I hope you all had a good lunch break. It's just a pleasure to be invited to speak today. Disappointing that I cannot join you physically today, but hopefully I will have other opportunities to visit your college in Nasik. Okay. Without spending too much time, I'm just sharing my screen, and hopefully that will work. Can you confirm that my screen is visible? Should be okay? Because I cannot hear any feedback. Yeah, okay, great. Thank you. So the topic of my presentation today will be focusing on in vitro testing of complex formulations. We are dealing with this quite a lot, simply because Sotax Pharma Services is a company which has been built about in vitro testing, and it's really within our DNA. We just celebrated, and in 2005 we were the very first company in the world specialized in that field, dealing about developing methods for dissolution and release testing. So we claim that that's for experts. All the people that you will see on the pictures are our employees, and the thing is, our goal is really to understand the properties of the formulation, and then to develop appropriate methods, because the method should be driven by the design of the product, the formulation. Okay. So at Sotax Pharma Services, we have 3 locations. One, the historical one, founded 20 years ago, was in France, in Orleans. In 2021, we opened our second site in Ahmedabad. Namely, it's called Ortiv, but that's a Sotax Pharma Services site. And more recently, in 2022, we opened our third lab near Boston. The goal is really to have a global organization acting locally for our customers in Asia, in Europe, Middle East, and Africa, and for the Americas, North and South. Our labs are dedicated to in vitro testing and are all GMP compliant. So we have a very strong history of customer audits and health authorities inspections. And we are registered as pharmaceutical sites for US and for Europe as well, which is quite unique. And what is very special is that we work as advisors for the industry, for our customers, but also for the health agencies. SOTAX now is very well known, it's a very strong name. And we are even doing consulting or organizing training for the health agencies staff. Myself, I organize trainings for the US FDA. Next month in October, I'm going to the Saudi Arabia FDA and so on, simply because they need our expertise. We are interacting with them, and this helps us to really be ahead and of our for our customers and ready to support properly both industry and health agencies. So I'll go quickly on these slides. These are all the services that we offer in R&D for method development, for GMP testing, including method validation, method transfer, stability studies, testing of commercial clinical batch, and support services, troubleshooting, investigations, training, or consulting. If you have any questions about that, do not hesitate to reach out to our contact at SOTAX, and they will find you the right person to address your request. That's the outline of my presentation today. We'll be talking a few words about really the basics of dissolution. I'm always including these few slides just for people to understand why is in vitro testing gaining so much importance in our industry, okay? And then I selected some cases of complex formulations. I'm talking about injectable suspensions. Other injectable formulations, you will see there are a couple of cases which will be addressed here. Combination products, and then a few words of conclusion, okay? Let's start with the basics and the fundamentals of dissolution. When we develop products, we are not developing tablets for dissolution vessels. We develop tablets for humans, for patients. And when you administer a dosage form to a patient, you have different steps. So the dosage form is first releasing the API. Once it's released, then it starts to dissolve, and only when it is dissolved, it can start to get absorbed, going through the GI tract membrane, for example, going to the blood, being distributed, and then you have efficacy and eventually safety concerns. And when we do clinical studies, we give a tablet to a patient. Usually we are measuring the concentration of drug into the plasma or into the blood, and these PK profiles, pharmacokinetic profiles, are characterized by 3 parameters. One is Cmax, maximum concentration that you can reach after Tmax. And the green zone here is the AUC, the area under the curve, and that is an amount. That is the amount of drug which is getting absorbed. And that is what we call the bioavailability of your drug. You may have a drug which is administered with a 100 milligram tablet, and you have only 15 milligrams which are getting absorbed. So the bioavailability is only 15%, but usually it is designed in a way that those 15 milligrams which are getting absorbed are sufficient to get the therapeutic effect. And the bioavailability of your drug is limited by the slowest of these steps. So it can be permeation, the absorption, and that's the BCS class 3 products. And when you have a BCS class 3 product, as a health agency, I'm quite comfortable to accept, for example, a generic for BCS class 3. Why? Because the bioavailability and the performance for the patient is essentially driven by the absorption. And the formulation itself, so the dosage form or the API dissolution, are not playing or not limiting the bioavailability. So there is no risk or limited risk that the formulation itself is affecting the bioavailability, and therefore less risks also for the agency to approve such product. On the other hand, you can have BCS class 2 products, which are highly permeable, so absorption is very quick, but dissolution is very slow. And then the dissolution API is on the critical pathway, and it's very important to make sure that the API dissolution or the API release are not limiting or affecting the performance of the generic versus the innovator. And for BCS class 2, dissolution is a critical step in the bioavailability of your product. When we drop a tablet in vitro in a vessel or in a flow-through cell, we are only starting with the dosage form and ending with dissolved drug, okay? But the tablet, when you drop it in a simple paddle instrument, it's not just dissolving, you have different steps. First, the disintegration, which can be driven by the properties of the formulation related to tableting or disintegrate or cohesive properties of the formulation. Then you have the drug release, which can be affected or modified depending on the excipient that you are using or the amount of excipients that you are using. And then only you have the dissolution of the API itself. And what is very important is to understand the relative importance of these steps in the dissolution test. Used to do dissolution testing on dosage forms, on the drug product. What we recommend sometimes is to do dissolution on the API itself, and to do so, we are using a technique called apparent dissolution. I'm not sure if the titles are really visible with this title, but I cannot move it, or maybe I can move it here. Yeah, sorry. The apparent dissolution is a technique which can be used for testing of drug substance of pure API. You have here it's a test which is described in the pharmacopoeia, and it looks like this. So hopefully here you will see, for example, testing of 2 batches of API coming from the same supplier. And we are always using this video because it's very clearly showing that the batch on the right-hand side is dissolving much faster from the batch on the left-hand side. And if you manufacture a tablet with this batch, you may end up with out of specification results. Why? Simply because the particle size are too large, dissolving slowly, and the problem is really related to the drug substance and not to the formulation, okay? And what we try to do, or what we recommend to our customers, is really to run or to compare the dissolution of the API with the dissolution of the dosage form of the tablet. And that can give you a lot of insights about what is critical. And I will highlight that in one of the case studies later during my presentation. That's a typical comparison of batches of acetaminophen, paracetamol, that we compared, having different particle size, and you can see that some of them are almost not dissolving, while the other are very different, really driven by the particle size of the API. Okay. So now coming to the main topic of my presentation, complex formulations. Dissolution testing started in the, let's say, 60s, 70s, and for a very long time it was really limited to oral formulations, so tablets and capsules. And the first reference that I found in the literature was in 2006 with Eli Lilly, who presented data showing that for injectable products, they observed differences in the bioperformance for patients, and that bioperformance was related to the particle size of your injectable suspension and they used in vitro testing to compare the batches and ensure batch-to-batch consistency. Okay. That methodology was also used later on by Sandoz, which is a generic company, and their site in Canada was very specialized in injectable generics. And here Daniel Abran was describing that they are systematically using in vitro testing to develop their new generics, to test them in vitro before going to in vivo clinical studies. Okay. And I want to illustrate that with a case study that we had for one of our customers. They had a successful bioequivalence in vivo with patients. When they submitted their dossier, they got a deficiency letter from the FDA indicating that the submission lacks dissolution data for the test product. So in all cases, the US FDA for injectable products is expecting to have dissolution data or in vitro data. Another statement, based on the information available to the Department of Bioequivalence, USP apparatus 4 appears to be more appropriate for dosage forms such as suspension compared to USP apparatus 2. That is very clear. That said, they were recommending to explore USP 4 as well as USP 2, okay, and to provide all dissolution method development, including selection of the dissolution medium, surfactants, if any, surfactant concentration, size of filters, and so on. So the customer came to us and we established a development strategy in order to fulfill the request from US FDA. In that specific case, the product was containing 2 active ingredients. One was in solution, was already dissolved, and the other, which was critical, was the suspended API. Okay, so we had the test conditions, trials with both USP4 and USP2, optimization of the selected method. So we selected an appropriate medium based on solubility. We selected appropriate filters, and then we tested the product with USP2 parallel instrument. It was 1 ml of suspension in 500 ml of medium and 25 RPM, which is the minimum speed that we can achieve with USP2. And here the profile, as you can see here, within 5 minutes, everything was dissolved. Okay, so here you don't have problems with the specifications, but a profile like this is useless. Why? Because everything is dissolving too fast and the method will never be discriminating. The role of a discriminating method is to be able to detect and to reject a bad batch, while here, all batches will probably always dissolve very quickly. So what we did is we used exactly the same conditions with the flow-through cell that is available in Nasik today, I think. And we applied 1 ml of our suspension here, 500 ml of the same medium with a closed system. And we showed that we used a flow rate of 8 ml per minute, which is an average flow rate. The standard flow rate are 4, 8, and 16, okay? And here with an average flow rate, we shifted from 5 minutes to 6 hours. With exactly the same conditions, the same medium, the same volume, and the same analytical method, we are shifting from 5 minutes to 6 hours. The reason for that is the hydrodynamic that we are offering within the flow-through cell. So from one dissolution instrument to the other, you can have very different hydrodynamic, and that is playing the major role for the discrimination. So now I have a profile for my drug product, the red one. My question is it a dissolution profile related to the API itself, or is it a release profile related to the formulation composition? So what we did, similarly to the previous video that I showed you, we tested pure API, non-formulated, with exactly the same conditions. So same amount of API, same volume of medium. And what we noticed is that The drug substance alone we tested that only over 4 hours. It was investigation, but we showed that this was much slower than this one. What is the conclusion of this? This profile is mainly driven by the formulation. And that is very good news for our customer because the API, they are buying it from an external supplier. And the good point is if the quality of the API is slightly changing, accelerating or decreasing, it's unlikely to affect this because this is driven by the composition of your vial because in the excipients that you have some wetting agents, some surfactant, and these excipients are mainly enhancing from the drug substance, pure drug substance, to the formulation. And this test on the pure API with the powder cell is not mandatory. It's not required by the agencies. But if you do it, then you are gaining a better product understanding. And if you have a problem on your product, if you have an OS, for example, you can very quickly investigate and make sure that you are checking in priority the batch master file for the drug product. And because you know that the API is unlikely to be the root cause for that problem. Okay? Now, if you go to the FDA website, there is a list of products for which FDA is recommending to use the flow-through cell or apparatus 2 for comparative evaluation by the agency. Okay. Other injectable formulations. In this chapter, I would like to talk mainly about liposomes, but this can be also covering nanosuspensions or emulsions. Okay, we have 2 papers which are very interesting, published by the University of Michigan and the University of Connecticut. Okay. And this paper, published in 2010 by Professor Diane Burgess and her student, Karl Bardwaj, is really the founding paper about testing liposomal formulations using dialysis membranes. Okay. The advantage of the second one, published by Anna Schwendemann, is that it was co-authored and co-founded by US FDA, which brings also some strong weight to this paper. Now, focusing on this first work by UConn, at that time, what they designed is a device allowing to use dialysis membranes, tubulins within the flow-through cell. And that was very successful with regards especially to the discrimination and the reproducibility of the profiles. Okay. Later on, this research device has been replaced by 2 tools. The first one is the floatalizer, which is then introduced in a standard flow-through cell, and more recently the microdialysis, which is also a modified dialysis membrane. Both are commercially available from Sotax, Sotax or Sotax Pharma Services. And just for you to know, Sotax has been, we are in touch and we just signed a research collaboration with the US FDA, and we will be working on this microdialysis technology together, US FDA and Sotax, in the coming months. So hopefully that will be published in the coming months. So just stay tuned on that, but you will be informed. So these are already commercially available since a few years now. I want to mention also another type of injectable products, which is subcutaneous or intramuscular formulations. And this is really something innovative. It has been released only this year, 2025. So it's one of the very latest innovative tools that we have. And this has been a collaboration between SOTAX and the National University of Singapore. It's Professor Matthias Wacker with his student David Lee. They worked on exploring the release dynamics of when you have subcutaneous drug delivery. One of the issues with injectable product is when you inject your formulation, SQ or IM, the formulation, the depot is entrapped into the tissue. And what we notice is when we test those products in vitro, depending on the size or the shape of the implants, you can have variability in your profile. So the idea is how can we mimic in vitro the physical constraints that our formulation is facing in vivo. Okay. And for that, they designed a new cell. It's called BioJect cell. It, as you can recognize, it's fitting in a flow-through cell, in a standard flow-through cell. And the trick is that the close cyst, but your formulation is entrapped in a cartridge here, which is made of agarose. And in the reality, it looks like this. And the advantage is that this cartridge made of agarose can be also complemented with collagen or hyaluronic acid to mimic the composition of the tissues in the SQ zone or into the IM zone, okay? And this has been largely explored here for insulin in this paper published this year. And the conclusion of this is that it's really interesting because it's showing a robust and highly sensitive methodology to evaluate drug release for those insulin formulations. The advantage is you can try to make the environment more biorelevant by using hyaluronic acid and collagen into the agarose biomatrix. And this allows to better understand the release mechanism which are underlying those profiles that we are getting. And this device called BioJect is now commercially available also. And we started already some work on that to have more case studies. And with different of our big pharma customers, they are really working hard on BioJect in order to have that used for all their injectable products. And probably the FDA is already showing some interest in that technology. Okay. Final type of formulations or product that I would like to highlight is combination products, where you have a drug, could be a very simple molecule like paracetamol, could be a more complex molecule like sirolimus or apamycin. And this drug is combined with a device. There are different types of device, even catheters, prostheses, and so on. And all that is forming what we call combination products. So a device releasing an API. And you have lots of intrauterine devices for contraceptive purpose. You have pacemakers. These are the leads for the pacemakers. Contact lenses which that you can put in your eye. And depending on the plastic or the polymer that you are using, you can put the lens and it's releasing all the drug within 1 hour to, for example, to induce mydriasis for the eye surgery. Or you can put it in the morning and release it, remove it at the end of the day after 12 hours, and you have local continuous drug delivery of an anti-inflammatory drug into the eye. So all those devices require in vitro testing. And I have 2 examples to share. One was very early research we did with the university in Limoges, where we tested bioceramics which are used for bone repair. And those bioceramics were loaded with an anti-inflammatory drug, which was ibuprofen. And we showed that we can have different profiles, very discriminating. And these are the USP4 profiles, but in the 2 papers, we compared USP2, USP3, USP4, and we showed that, and even a more innovative tool, which is called the T-cell for diffusion testing. The conclusion of this work was that in vitro, we had profiles ranging from 30 minutes to 30 hours. And to know which one is the more relevant, we need in vivo data just to be able to see which in vitro tool is better correlating with in vivo. So the final judge, I'm always mentioning that we can have very sophisticated in vitro tools. We should always remain very humble, because despite the sophistication of those in vitro methods, it's still very simple compared to the complexity of the human body. Okay. The second case, and here I will go a little bit more in details, is for intravaginal rings used for contraceptive purpose. We had a feasibility study that we did for our customer based on a paper published by the University of Greifswald in Germany. And here the ring was containing 2 active ingredients, and we tested only one of them using UV measurements. UV measurement was very interesting because it's allowing us to have very frequent measurements. So every single point that you see here is a UV measurement of the profiles. And you see that on day 1, we had a burst effect. Then on day 2, day 3, day 4, it's very consistent. And then we extended to day 5 and day 6, and they're always very consistent, okay? So over 24 hours, we have the burst effect as expected by the design of the formulation, and then very consistent daily amount of drug release. What we did is we were using the flow-through cells. So the diameter of the cell is 22 millimeter, but the rings are about 4 to 5 centimeters. So we had to twist them, to bend them, to have them fitting into the cell. What we did is we tested different positions of the device, of the ring inside the cell. And you see that over 24 hours, we had very reproducible profile. And this was highlighting that the position of the device itself is not playing a role, and that the release mechanism is mainly driven by diffusion, not that much by the erosion or the hydrodynamics, because we also tested different flow rate and we didn't see a difference. So now the next steps, once we have reproducible profile, is to go for discrimination. So we ask our customer, can we have samples having different release properties? So here we switch from UV measurements to UPLC in order to quantitate both compounds, X and Y. And we tested at 37 degrees, so I'm showing only one compound here. And you see that we have a very good discrimination between good batches and bad batches. And as you can see, if you remember at the very beginning, we made one test from day 4 to day 6, simply because you see here, you have the weekdays, and this is the weekend, weekdays, weekend, because here we are not running over 24 hours anymore, it's 21 days, which is the in vivo duration for the use of the ring, 3 weeks. And here we are having a very good discrimination, but it's 21 days. That's a very long profile. And because we have a release mechanism driven only by diffusion, we wanted to accelerate that. And the only way is to use a higher temperature. So it's not physiological anymore, but that's not the goal. It's not to mimic the physiology, it's to have consistency and discrimination. And here we can have the same amount of drug released in only 12 days. In only 12 days, as you can see here, and discrimination is still there. Now, can we go a bit further, 55, and here you can see that we need only 6 days to achieve the same amount, but we don't have any discrimination anymore. So overall, for the conclusion, we have a real-time method, 21 days. We have an accelerated method for 10 days, or 12 days, And then this one was too much accelerated, and we don't have discrimination anymore. And the conclusion when our customer discussed with the health agency is the health agency wanted the real-time method to be used during the development, and this would become the QC method. Since then, we used the method has been fully validated, ICH, and we tested all the stability samples, and the product is now approved and marketed. It's fully commercial, running with the 10 days QC method. Now, jumping to the conclusion, I think I'm on time, opening the curtain on this conclusion. What is really important to keep in mind is we have more and more complex dosage forms, and the goal is not to have something fancy for researchers, is to have something useful for the doctors to treat the patients, okay? So those formulations are getting more and more complex. If you compare a simple limited release tablet with a combination product, there is a whole world of complexity in there. And because of, as I mentioned at the very beginning, the testing method should be driven by the design of the product of the formulation. And for this, it's really important to adapt the operating conditions in order to understand the critical quality attributes or the critical process parameters which can be affecting the bioavailability, okay? And for that, modern techniques like the flow-through cell, for example, are more flexible, more versatile, and allows to better match what we are looking for, what we are trying to understand. The goal of dissolution is not to obtain a profile for your dossier, is to have a profile which is meaningful and which provides you knowledge about your product, okay? And with this, I would like to thank you for your attention. I would like to thank again the organizing committee for inviting me and giving me the opportunity to be part of this conference today. Okay, thank you. Thank you. I'm not sure if you are planning to have questions now or later. Quite flexible on this.
Anyone have any question? One question. Any question from the audience?
Right.
Good afternoon, sir.
I cannot hear you. I'm sorry.
On Grand Mercy. On Grand Mercy, Amu Samir Hadochi, Pur Amar Partaze, Unvar Parsio Si Parsio. Unvar Parsio Si Parsio. What I've just said is a thank you note to Mrs. Samir Hadochi in French. I just tried that. I'm not very fluent in French, but what I'm trying to say is a big thank you to Mrs. Samir Hadochi for sharing such a valuable insight on the complexities of the in vitro testing. Your expertise and practical approach have shed light on the challenging and the innovation shaping pharmaceutical formulations today. Thank you, sir, for joining from this far and being a part of our international conference. Thank you.
Now, I would like to invite Dr. Tambe sir to felicitate Dr. Padma Devarajan ma'am on behalf of Samir Hadochi sir. Sir. Yes, yes.
He is able to hear you. He cannot hear you.
Thank you, sir. It is my privilege to introduce Dr. Padma Devarajan ma'am, a distinguished leader in pharmaceutical sciences and currently the president of the Society for Pharmaceutical Dissolution Science. Dr. Devarajan ma'am is serving as an advisor and chair professor at the SIES School of Pharmaceutical Sciences, Mumbai. She retired as a dean, researcher and innovator and professor in pharmacy at the Institute of Chemical Technology, Mumbai, where she also headed the Department of Pharmaceutical Sciences and Technology and served on the boards of governors.
Thank you very much. There are no questions, so you can leave. Thank you very much.
Okay, great. Thank you so much, and have all a good afternoon. I'm sorry. Bye-bye, everyone. Namaste. Thank you.
Her academic and research contributions are exceptional. She has authored over 100 reviewed publications, hold several granted patents, and has successfully licensed technology to industries with products commercialized both in India and Europe. She is also the editor and author of 2 books on targeted drug delivery published by Springer. Dr. Devarajan ma'am has been associated globally, having served as a board member of the Controlled Release Society, USA, chair of the Global Young Scientist Mentor Protege Program, and the editorial boards of leading international journals. She also represented India at the ISO Working Group on Nanotechnology Safety. Currently, she has been appointed as the chair of the National Innovation and Startup Policy 2024 Revision Committee by the Ministry of Education, Government of India. She has contributed widely in the field of nanotechnology. She is a fellow of the Maharashtra Academy of Science, a life fellow of the Indian Chemical Society, and the recipient of several prestigious awards, including the AAPS Distinguished Educator and Researcher Award, the Outstanding Woman Scientist Award, among many others. We are truly honored to welcome such an accomplished professional today. Please join me in welcoming Dr. Padma Devarajan ma'am. Now I request our honorable guest to kindly commence the session.
Thank you very much. I would once again like to thank Dr. Tambe and the entire team for giving me this opportunity to be here. And I think it's a very good sequel that I'm following Samir instead of having come before him as it was planned originally. And one very important take-home message from Samir, which was almost the last line that he said, that dissolution should not just be a line or a curve. It has to be something with a lot of meaning. And the meaning comes based on how you plan the dissolution. So it's all integrated into understanding dissolution and then coming up with an appropriate solution. So while Samir talked to you about injectables, I'm now going to take it further into a very important set of delivery systems that is being worked on extensively in most of the institutes in India. I'm not very sure if MGV Pharmacy College has also started, but I'm sure you will start if you haven't. So I'm going to talk to you today about the testing of nanosystems, the various challenges and the opportunities. So first, what is nano? This is just for people who probably would not even be able to visualize what is nano. So what are we talking about? We are talking about a size that is less than 100 nanometer. And this line, if you see here, the red line, is that 100 nanometer line. So anything less to the left of that is nano. However, when we talk of nanomedicine, that has been expanded. So it has been expanded, it has been expanded to almost 600 to 700 nanometers. So when we talk of nanomedicine, it's not always that a size of 100 nanometer is going to be useful or less than 100 nanometers. There are many occasions when we need to be at 200, 300, 400, but an upper limit is about 600 to 700, beyond which one is taking a risk of occlusion of blood vessels. But when do we need this larger size? When we're looking at intracellular infections. And if I'm looking at cancer, I would look at the lower size. So it was quite a challenge when in the earlier stages when we would try and write proposals to defend that we needed a larger size, because you'd be defending in front of a typical science community who's not particularly pharmacy oriented. And it was a huge challenge, but today everybody has understood that nanomedicine does not mean size less than 100, as you will see in typical definitions. It means a size that could be more than 100, but generally about 700 or so is what we have found is a size that can still be safely administered without challenge. So here is just a pictorial view of what we are looking at when we talk of nano. And what do we get when we look at nano? We get a very huge expansion in surface area. So if you look at those 3 blocks, you will see the expansion. It's lakh square centimeter expansion. So the expansion in scale, when I convert something from a 6 square centimeter to nano, can you see it goes to 6 lakh or 60 lakh square centimeter. So we're talking of huge surface area, which means we're talking of huge energy. So when we talk of huge energy, you know energy can destroy. And that is one huge concern, that when I go into the nano state, can that walk into my cells and destroy something inside? So that's one of the biggest challenges of nano today. So let's come to nanomedicines now. We just heard the word liposome from Samir. And here is a picture that kind of summarizes most of the nanomaterials and nanoformulations that are being worked on. So in case of nanomedicines, what do we need to focus on? One very important, of course, is the size, which I mentioned. So should I continue with the laptop here? Okay, the other is, of course, surface area. The third is material. What is the nanomedicine made up of? And the fourth is a very important property, that is the shape. Because when we talk of nano, there is an assumption that it's round, that is spherical. But today you have a variety of shapes that are being used for very many different specialized applications. So we can look at all these when we talk of nano. So now this is various formulations, nano formulations with reference to size. So the bar in the center, you can see starts at 1 nanometer and goes to 1 micron. That is 1000 nanometer. And you will see that carbon nanotubes, fullerenes, dendrimers are all almost very tiny, less than 10 nanometers. As you move up, you will see the micelles, you'll see the neosomes, you'll see the liposomes, you'll see the SLN. And all of them, if you see, they're on the border of 100 and they move a little right and a little left. So you can be maybe 50 nanometers to maybe 700, like I said. So with all these formulations, which are mainly lipidic formulations, for nanomedicine, they are ideal formulations because lipids generally are biocompatible and very, very safe. And they're most of the time biodegradable and metabolized by the body. So we are going to look at nanocarriers. When we talk of nanocarriers, if we are talking of dendrimers, carbon nanotubes, etc., they are really tiny and even more difficult to handle. What I would be focusing on is something that's maybe 80 to a little more than that, like about 500 or so. Okay. So now, why are nanoformulations important? Why are we talking about them? We have success stories, which you see at the bottom. These are two of the very major success stories. But as of September 2025, if you see on the right-hand side, about 100 nanoformulations have been approved for therapeutic use by major regulatory agencies. But if you look at that number 100, it's nano compared to the number of formulations that are approved by regulatory agencies. So you can see that although nano initiated in 90s, which is almost for 30, 40 years now, we have only 100 nanoformulations approved. So it's not a big number. It's yet a small number, but it's a very promising number that says there is a good chance that the growth could now be logarithmic because we understand much more about nanoformulations. Today, the understanding is increasing in leaps and bounds, and therefore there's a good chance of it going logarithmic. The advantage being targeted delivery, enhanced efficacy coupled with decreased systemic toxicity. And if we talk of that one drug, Doxil, which is on the left side, which is liposomes of doxorubicin, the liposomes have almost made the challenge of cardiotoxicity of plain doxorubicin solution 0%. So it's a very, very effective solution, but it's a very, very expensive solution. It's a high-tech, not readily scalable product, despite being commercial. So it's a huge challenge for industry even today. So we do have still a long way to go before that 100 keeps moving at a logarithmic rate. So coming to nanosystems, now that we already have about 100, FDA is gearing up to control, bring in regulations. So you need this during research, you need dissolution testing. I'm not going into all the other things. And why dissolution? We've had enough talks in the earlier part of the day. So we need it for research, we need it as a quality control tool. We may also need it for bioprediction. And today we do not have standardized in vitro dissolution methods for nanosystems, and there is an urgent need. Just like Sotax is working on subcutaneous, intramuscular, and in fact the BioJet that you saw, it's probably the first time that Samir Aduchi is presenting this in India in any conference. So it's a real new development that all of you have got to see much earlier than anybody else, even in big tier cities. So let's go through how one could actually look at this development of dissolution for nanosystems. Now, these are the typical dissolution systems that you're familiar with. And for those who had not seen USP4, you've seen it here today. You saw it in Samir's talk just now. So you have USP1, you have USP2, and you have USP4. And in all, what you're seeing is a tablet dissolving. So for oral dosage forms, these are very well suited. And they're being worked on also for other dosage forms. So now, so when I'm talking of nano, why is it that I can't just adapt USP 1, 2, and 4? One problem is the size, which does not allow separation of my drug from the dissolved drug. Because whatever filter I use, the drug is going to come. Because you maximum use a 0.22 micron filter, which is a sterilization filter, and your nano is sometimes 100, 200, it's going to come through. So separation is very, very difficult. So you need complex methods. And many times the system is so complex. So maybe it's a liposome, maybe it's a lipidic particle, but it is designed to work, say, intracellularly in the RES or intracellularly in the cancer cell. So whether dissolution would give me any indicator of what's going to happen, it's not clear. So there are challenges. There are many systems that are developed for maybe pH or temperature sensitive release. So that again is difficult to mimic. And when you have a program release system, setting up a dissolution test becomes even more complex. So what are the methods that have been evolved? I'm taking you through the evolution of dissolution for nanosystem, where it began and where we are today. So the first methods are the sample and separation methods. Then came the membrane diffusion dialysis sac methods. You saw one in our earlier presentation by Samir. And then you have other methods like microdialysis, dynamic dissolution, two-stage reverse dialysis, etc. I will show you some insights into these. This pointer, okay. So now, if I look at, I think it's jumped. No. Okay. Now let me look at this. I can use the official apparatus and modify it. So here you have something that is a modified USP1. It's not the typical basket. It's a membrane kind of cell. And then you have the continuous flow through method, which Samir just showed us. Now in USP1, the challenge one faces is a limited volume and you do not have a sink condition. In USP4, the sink condition problem is totally overcome because you can keep using as much volume as you want and you'll have a fantastic demo towards the last session by Mr. Suhas Yevle. So you would understand that much better. Okay. Now, what is a sample and separation method? I add my sample to, say, a USP1 or a USP2. When I withdraw an aliquot, I have to ensure that my nanoparticles are not in that aliquot. So you need specialized filtration techniques like ultrafiltration. Or you need a specialized technique like ultracentrifugation. And when you are trying to do this, you have to take out a sample and then you have to return the sample back, or whatever you have withdrawn in that sample, what is not dissolved, if it's not put back, it's going to change your profile, your quantitative metrics is going to change. If you try to put it back, it brings in a lot of errors. So there are challenges when you have this sample and separating method, particularly because you don't have online separation techniques as yet. The other method The other method is pressure ultrafiltration, or you can use syringe filtration. Again, the filters and syringes, you don't get 100 nanometer filter, etc. And even if you get, they choke very rapidly. And they are very, very unaffordable for research in academia. So that's not a practical solution at all. I think I have to find a solution for this first. Okay. So these are the problems of sample and separation method. So there's a difficulty in separation. The second important challenge is the time taken, because ultracentrifugation, ultrafiltration, etc. don't happen in minutes. It may take 10 minutes to 30 minutes. And during that process of separation, drug release continues. So you're bringing in a lot of artifacts into your values that you're measuring. Because suppose you have centrifuged one sample for 25 minutes and the other you go for 35 minutes, you're not sure you're going to get reproducibility, etc. So there are challenges with this method, which was among the first methods that were studied. And then they came up with this continuous evaluation, that is dynamic dissolution. And this was based on ionization and using electrodes, so that when the drug is released and it's ionized, it would automatically get detected. But now you know that when I have a drug, it has to be ionizable for use by this method. So that itself brings in a lot of limitations. But yes, if you have an ionizable drug, this is one of the practical methods that can be used. It's almost like online testing. As you have more and more drug release, you will be detecting more and more of the ions. So today, based on the difficulties with the sample and separation method, which was the first method, the dialysis methods became more popular. So in dialysis methods also, there has been a lot of efforts that have gone in before you saw what the USP4 shows. So when I am relying on a dialysis method, very important parameter is the agitation. Because if I have a membrane, I could have something static inside the membrane. So if it is not stirred, there could be a challenge, there'd be a diffusion layer developing, and this may not be reproducible. So you need to have the agitation condition optimized. The donor medium volume should be 6 to 10-fold less than the acceptor media volume. That is very, very important. That may not be a very difficult thing to achieve. Again, when I have a membrane, the molecular weight cutoff of the membrane must be minimum 10 times more than my drug molecular weight. So if my drug molecular weight is 500, my dialysis MWCO should be minimum 5,000 or more. If you can go to 10,000, 20,000, it's even better, so that the membrane is not limiting the movement of the drug that is released from your system into your acceptor medium. Okay, so this is a pictorial depiction. So here you have a dialysis sac. So you start the dialysis, you have a high concentration gradient. At the end, you can see the drug is all come out on the right side. And once there's equilibrium, you can't, so you have to manage sink condition. So you have to ensure that there's going to be sink condition in selection of the volume of your sample and in the volume of your acceptor medium. So this is a simple thing, but if you look at the sac, theoretically it can remain that way, but practically it would not retain that shape at all, because when I fill water into a soft sac, it's going to acquire any kind of shape, so the surface area would keep varying depending on how I put it, what is my agitation, etc. You had a reverse dialysis sac where you put your sample outside and your medium inside, but then the medium volume is going to be much, much smaller than sample volume here. So that again is a challenge. And then one looked at a side-by-side dialysis membrane where they put the nanoparticles into that glass tube with a dialysis membrane at the bottom. And you put your nanoparticles there and you did a side-by-side. So it could be a modified USP. The basket is replaced. But even this, it was found that it was not giving the kind of reproducibility and the kind of predictability that one would actually like with any pharma system. So where did these developments begin? Way back in 1968, the dialysis bag was used. It was a stationary basket rotating paddle method. And then the next time they had a dialysis bag with a rotating paddle, and that bag was stirred by a magnetic needle. So you had a needle inside the bag. So a lot of things have gone in. This is just to tell you that you should not, if you start thinking differently, make sure all this you have gone through and you're not repeating history. Here you had the USB basket turned around and modified into a dialysis cell, and that basket kept moving round and round. So these are all, in the early 60s to 90 developments, and they kept moving on and on. Okay, so if you see here, this is another kind of equipment that came up. This was a continuous flow cell with the dialysis membrane. So this was adaptation of dialysis with a continuous flow cell. So if you look at this, the flow cell is expanded here, but that red line takes you to that little flow cell there. In A, you have the release medium. In B, you have a pump, and you have that pump continuously circulating the medium. So this was before the USB4 came in, a much cruder version of the same thing. So this was quite recent in 2015. You can see it was published in APS Pharmaceutical 2015. So this was the beginning, but this was not considered good enough, and that's how you have further modifications. So I should do this? Oh, please tell me that earlier. Okay, so now you see this, the next one. So you have a basket and you have that basket modified into a dialysis cell. So this was another method. This was done in 2013 and with, oh, perfect. So here you see this. I'm showing you some data. You look at this here, they're looking at the effect of pH and of enzyme. The red lines are the effect of pH. So the triangle is 7.4 and the square below in red is pH 4. So you see that there is good discrimination in pH. You can see the discrimination in the test. And similarly, in the presence of trypsin, you see there is a difference. At the same time, the pH effect is also seen there. So like Samir just told us, in the dissolution test, you need to have a test, you need to have a release pattern, you also need to have discrimination. But when we look at those, the basket modified, etc., there are certain challenges. So one is lack of adequate agitation inside the membrane. For instance, even if you take the size of a USB basket, it's almost 1 centimeter dia, and that is a huge diffusion layer thickness. So that would need agitation, which is not there in the typical USB. There is violation of sync condition when I'm using USB 1 and 2 because I can use only up to a liter of medium. Maybe with a large vessel, I can go to 2 or 4 liters, but then I may be diluting too much. The reverse system causes too much of high dilution, which I showed you earlier. And this diffusion is one of the biggest challenges, which gives erroneous results and, gives you huge error bars. So one approach is, yes, what Samir said. So I'll go through this very fast. This is a USB 4. He's shown it to us. Please, there's the same picture that he showed. But very important, look at the left side. That's the dialysis sac with the nanoparticles. Now, in this dialysis sac, putting that membrane, was very tough in that adapter because we have used it thanks to Sotax. But it was very, very tough to put the membrane, and that was among their first adaptations. So although that method was used to investigate drug release from microspheres, for nanoparticles, it became difficult because they were very, very tiny. They were clogging the filter, resulting in pressure buildup. And they would also pass through the filters. So they came up with a dialysis adapter, which we saw. But with the dialysis adapter, there was a challenge in using it. And therefore, today what you have is this kind of a dialysis cell. This is called a floatalizer. So, this is actually an expanded version of that adapter only. So this, you can see that membrane, now it looks very neat. But getting it around very neatly was the biggest challenge. So, here. So now let's go to some data. So here, this is a simple USB 4 dissolution. The blue line right near the y-axis you see is salmon calcitonin solution released almost immediately. So this told us that we can use that dialysis membrane because it doesn't limit our drug. And then we got a good sustained release profile with some salmon calcitonin nanoparticles that we had made in the lab. Next, we compared USB 1 with USB 4 and we used a dialysis sac method. So here, if you see that sac, that's how it stands. It never stands as straight as I showed you in the earlier picture. But nevertheless, we put it into the basket. And when we did a disso, the red line is USB 1 and the blue line is USB 4. Now, very important, what we noticed with the red line, that's USB 1, is a huge standard bars. And that's because that bag goes in any form, in any shape, and that affects my surface area. So this says that that's not really a practical method. Even if you feel, I don't have anything, can I do it? It's not the right thing to do. Because look at the error bars, they're really, really high. And when I use USB 4, there's a significant lowering in the standard deviations. Here we are looking at different parameters using USB 4 and effect of pH. So on top is ALF, that is artificial lung fluid. And this was for rifampicin nanoparticles, which we had developed for lung delivery. So as you move from left to right, you see effect of flow rate on both top and bottom. And you see that as flow rate increases, the release rate also increases, particularly as you move from 8 to 24. But in the 16, in one case it matched that, in one case it matched this. So when you have your own system, you would have to understand what is the best flow rate to use. But in all the three cases, if you see, at 16, we got good discrimination and we did not get good discrimination at 24 and we got very slow release at 8. So that is also becomes one important criteria to select a parameter during dissolution. And in both if you look at the data comprehensively, you will see that the release at pH 4.5 was much higher than at 7.4 and our particles were designed to give such release. This is another data that tells us when we use USB 4, our polymer B is a hydrophilic polymer. Okay, and polymer A is hydrophobic. So as we increased polymer B, when I go from left to right, the release profile kept increasing, the release rates kept increasing, and when I look at pH 7.4 versus pH 4.5, look vertically, I find that release is much faster at 4.5, as we had seen earlier also. Now this is a comparison of USP 1, USP 2 and USP 4 that we undertook. And if you see there, what we found is in terms of discrimination, etc., USP 1 and 2 were discriminating the drug from the formulations, but the standard deviations were too large. When we came to USP 4, again, a significant finding was the standard deviation being low, saying that we need to further optimize this method, or rather this process, to develop our dissolution method finally. So what we have here is a simple summary that says USP 4 with the adapter or the floatilizer is a practical method for evaluating nanosystems. Now, the major challenge with USP 4.1 is the apparatus itself is very, very expensive compared to 1 and 2. USP apparatus 1 and 2 are, I think I'll leave it to the SOTAX team to tell you the cost, but I don't know. But I know one thing, USP 4 costs more than 1 crore. The adapters you use are throwaway, and each adapter used to cost 5 years ago 700 to 800 rupees. Today, I don't know the cost. Maybe it's gone to 1,000. 1,500, right? So today each adapter, so you can imagine 9,000 to do one dissolution, 6 replicates. So again, the cost is too much. So we started looking at other things. So in that process, what did we also find in literature? So we found that somebody had made an adapter for USP2 called the Pharma Test Dispersion Releaser. This is now commercialized, it's available in the market. Now this particular releaser, look at the red part is a dialysis membrane, and inside that they have an agitator. Outside that they have an agitator. So it's a very complex kind of thing, and that apparatus also is not really very affordable. It's again very, very expensive, although they claim that the results from the apparatus are very, very predictable, reproducible, etc. So it consists of a donor and acceptor. The donor is critical and contains a sample, and it is surrounded by a dialysis membrane. Now, inside the donor compartment is a small paddle. You also have an external magnetic stirrer. I don't have a pointer, otherwise I would have shown you all that in the picture itself. So this design is very, very complex. So we did look at this design, and just to show you how complex it is, you can look, it's patented. So if you just look at Pharma Test, you will find the patent, and those interested can take a good look at it and understand the Pharma Test Dispersion Releaser. But look at their data. Their data is very good. They hardly get any standard deviations, etc. But then there are challenges, like I told you, of cost and other things. So now I'm going to present you a dialysis adapter, which we have designed in our lab for USP2. So it's as simple as this. It's a clip with 2 holders, and it's hung on the paddle the way you see on the right side. Okay, so it's just hung on the paddle. And what is hung, the bottom holds the dialysis membrane. I think I have a better. So the dialysis membrane is here. This is the clip, and this is what helps it hang on the paddle. So this we could do very easily. We actually started by using paper clips to design it. Then we've done it by 3D and arrived at this. So if you see this, it's a simple sac using a dialysis membrane. The amount of dialysis membrane you need here is also very limited compared to the USP4 dialyzing adapter. In fact, dialysis membranes are also expensive. So the less you can use, the better it is. So that's again one very important thing. Now, Samir mentioned the word hydrodynamics, right? So when I use USP2, that hydrodynamics is established at 50, 75, 100 RPM, etc. And that's how it's come into the pharmacopoeia. Now, we looked at the hydrodynamics with and without our adapter. And if you look at the colors on the left and the right, you hardly see any difference. So this was all modeled with the support of some engineering students at ICT. And we saw that the adapter is not changing the hydrodynamics. So this is again a very important finding. The other important fact about our cell is, you saw the dialysis membrane, it's spread out. It's very, very narrow. The thickness of your sample would be 1 to 2 mm only and not 10 mm. So 1 to 2 mm totally takes off the diffusion layer thickness challenge, and you don't need a stirrer inside the dialysis membrane the way they needed it in the pharma test apparatus. So that's again one very important finding that we have. And then we look at our profiles. So this is a USP2 dialysis bag where we just tied the dialysis bag and put it into it. And that was USP2 with our adapter. We've done this for lipidic nanoparticles of the drug favipiravir. So if you see, the standard deviations are so low, like USP4. So this told us this looks very, very promising. Now it's refusing to work even here, or has it changed? No. Oh. So now again, USP dialysis bag and USP novel dialysis adapter, we find that standard deviations are very low. We are getting good discriminations also. So the dialysis bag is not taking off the discrimination that you see. You see those 3 profiles that are separated out. I'll just move ahead. So what we have here is USP2 with a novel adapter. We filed the patent and the patent is also granted for that. And I'm very happy to say that Sotax is actually in discussion, or we are in discussion with Sotax for this dialysis adapter. So we are hoping that their next innovation in 2026 would be this dialysis adapter with USP2. Because it was for USP2, they got interested, because right now they only have it USP4. And they find that in many countries, USP2 is the predominant instrument used across industry. So first getting them to buy USP4 and then changing to using the dialysis adapter may be more difficult. And Indian industry is rapidly moving into nano also. So therefore, they have shown interest, and we're already signed a CDA and we're already sharing data with them. So a major advantage of this is it's adapted to USP2 without changing the hydrodynamics, and cost is again a very important advantage. Making it is very simple. It can be done by simple 3D printing. You don't need a very complex kind of engineering for this device that we've developed. So with this, I'd like to thank Sotax India, because all the USP4 testing we did was with Sotax India's support. This is the Institute of Chemical Technology from where I come. It's located in Matunga, and we have 2 campuses now. One in Aurangabad, in Jalna, near Aurangabad, and one in Bhubaneswar in Orissa. And this is part of my vibrant research group, and one of them is sitting right here, who's going to follow me and talk to you, Pooja Todke. So it's thanks to students that research really grows, it becomes exciting. So to all the students here, I must say, it is you who will make a difference. We teachers are here only to support. But if students are not enthusiastic, teachers really are at a major loss. I've been very fortunate to have very good students across my career. This is the institute where I have now joined, the SIES School of Pharmaceutical Sciences, which is in Navi Mumbai. So with this, thank you very much. These are 2 books that have already come out, and our 3rd book should be coming out hopefully by the end of this year itself, which is on receptor mediated stimuli responsive nanoparticles for cancer therapy. So thank you very much.
Anybody with the questions? Your perspective have broadened our horizons. A heartfelt thank you to Professor Padma Devrajan ma'am for so brilliantly unpacking the complexities surrounding dissolution testing of nanosystem. Your expertise and deep understanding of cutting-edge area have not only broadened our perspective but also sparked important questions and ideas for future research and development. Your ability to bring scientific details with practical relevance is truly inspiring. We are privileged to have benefited from your vast knowledge and leadership in this field. Now, I would like to invite Professor Dr. R.A. Patil, ma'am, who is the academic supervisor of PharmD, to felicitate Dr. Padma Devrajan, ma'am. I request ma'am to please come on the dais. Yes, thank you. Thank you, ma'am. Ladies and gentlemen, let's move forward with our next session, featuring an emerging researcher whose work is making significant strides in advanced drug delivery system. It gives me a great pleasure to welcome Dr. Pooja Todge, a promised scientist and innovator from Institute of Chemical Technology, ICT, Mumbai. Dr. Todge holds a PhD from ICT, Mumbai and is currently a postdoctoral researcher at the Lithuanian University of Health Science, Europe. Now I request Laiba Sayed to please introduce the guest.
It is my great pleasure to introduce Dr. Pooja Todke ma'am, a distinguished pharmaceutical scientist specializing in nanomedicine and targeted drug delivery. Dr. Todke ma'am earned her PhD in pharmaceutics from the Institute of Chemical Technology, Mumbai, where she developed a safer, cost-effective nanoformulation of amphotericin B. She also holds an MS from Nipur, Ahmedabad, and is currently a postdoctoral researcher at the Lithuanian University of Health Science. Her extensive contribution to the field include a patent and publication in high-impact journals like the Journal of Controlled Release and RS Pharmaceutics. Dr. Todke ma'am has received over 20 national and international awards, including the SPDS Young Researcher Award and multiple Best Oral Presentation Awards. Please join me in welcoming Dr. Pooja Todke ma'am. I now request our honorable guest to kindly commence the session. Hello. Very warm good afternoon, everyone. It is prestigious and honored to be speaking at the international conference. And I would like to thank the MGVS College of Pharmacy and the SPDS. So myself, Pooja, I am doing currently postdoc from the Lithuanian University in Lithuania, Europe. And today I am going to present my PhD research work, which have presented in a DRPI competition. So this is recognized and awarded me as a Young Researcher Award. And international level, it is got second prize. So the title of this presentation is Amphotericin B Nanoformulation Insta-MB
tracking safer monomeric AMB release. So as many of you know that amphotericin B is a gold standard treatment for visceral leishmaniasis. So there are two types of formulations are available in the market. The first, conventional AMB therapy, which exists in aggregated AMB, and that's non-specifically interact with the human cholesterol. So thus causes liver and fetal kidney toxicity. So to overcome this challenge, the issue of toxicity, the lipidic AMB nanotherapy came into picture, which exists in super aggregated AMB, which also called as a monomeric depot, which releases the monomer slowly and it specifically interact with the fungal ergosterol and thus decrease toxicity. But nevertheless, there are many challenges are associated with a lipidic AMB nanotherapy, that is scalability, stability, and manufacturing complexity. So that's why, therefore, we are presented that InstaAMB, a safer amphotericin B nanotherapy prepared by facile technology. So we have initiated that is InstaAMB development by screening lipids by using molecular operating environment software. For this molecular docking study, we have selected the lipids which are containing shorter chain fatty acid to the longer chain fatty acid. So you can see on the left side, the shorter chain fatty acid show incomplete wrapping, which proposes the aggregated AMB. While in case of on the right side, you can see the fatty acid chain containing excipient show complete wrapping all over the amphotericin. This proposes the super aggregated AMB. Further, we have also demonstrated the interaction of the InstaAMB with cholesterol and ergosterol by using molecular dynamic simulation. So for that, we have used InstaAMB lipid complex, control liposomal AMB, and control aggregated AMB. So you can see InstaAMB doesn't interact with the cholesterol, and you can see on the right side there is an extensive interaction with the ergosterol. The similar pattern was observed with the liposomal, which is a more safer form of the amphotericin B. So this indicates there is no interaction with the cholesterol. This indicates the safety of the formulation, and extensive interaction with ergosterol indicates the efficacy of the formulation. Now you can see here that at the control, that is aggregated AMB, as I told you, that is a very toxic formulation. So you can see at the cholesterol, there is extensive interaction was observed and limited interaction with the ergosterol. So this indicates the aggregated state toxicity. So after selecting the novel lipids, we have prepared the InstaAMB by using in situ nanotechnology. In situ nanotechnology is a perfect example of technology breakthrough. Why? So InstaAMB is a simple solution of amphotericin B and excipient in a pharmaceutical acceptable vehicle. When at the patient side, just withdraw it by using a syringe and just add it into the dextrose injection 5%. So you can see the instantaneously generation of AMB loaded lipidic nanoparticles. Now this InstaAMB is ready to inject. So it contains more than 95% AMB in trap. The images revealed at 80 nanometer in size, and the PDI is 0.340. And importantly, this InstaAMB is existing super aggregated AMB. So what is objective in the perspective of dissolution? We have used very novel application of a dissolution testing in a tracking safer monorelease from the InstaAMB by using simple UV spectroscopy. So what is the principle behind? That is, you can see here the three peak. The first peak of the UV spectrum, you can see there is a super aggregated AMB which between the 321 to 325. It is also called as a monomeric depot. The second peak, that is aggregated AMB, exists in between the 330 to 350 nanometer, and third peak, that is monomeric, between 405 to 450 nanometer. So how we have conducted the in vitro release for the InstaAMB? So we have used two dissolution apparatus, USP2 and USP4. So these are the conditions we have followed for the USP2 and USP4 to check the release of the study. Since the InstaAMB formulation is a parental intravenous formulation, so we have used simulated intravenous parental fluid, that is Cray-Bringer buffer PA 7.4 with a 4% albumin. So this is also InstaAMB is targeted to the leishmaniasis, that is this leishmania parasite specifically is reside into the lysosomal compartment of the cells. So that's why dissolution in this medium is necessary. So we have used simulated intracellular lysosomal medium, that is artificial lysosomal fluid, pH 4.5 with a 0.2% between 20. So marketed liposomal AMB formulation was used as a reference here. So coming to the major objective of the tracking monomeric release. Here you can see in the USP2 apparatus, we can see that in the KRP media shows very low release as compared to the ALF. The rapid release was observed and you can see the standard deviation is very high. Coming towards the tracking by UV visible spectroscopy, the first at the initial time point, at the 10 minute, there is a liposomal AMB and InstaAMB shows super aggregated AMB. Over a period of time, this shows the monomeric AMB release and which is confirmed through the dissolution media by UV visible spectroscopy. Next, the USP4 apparatus result, you can see very low standard deviation was observed compared to the USP2. You can see here and here the complete release was observed as compared to the USP2 and a faster release in ALF compared to the KRB, which is augmented intracellular drug release. And in case of the tracking by UV visible spectroscopy, you can see at the initial time point, the super aggregated AMB was observed, while over a period of time, at a 6 hour, the monomeric AMB release confirmed in a dissolution media by UV spectrum. So the toxicity of the amphotericin is a major challenge. So the in vivo, we have used in vivo zebrafish toxicity study. So it is necessary to check the toxicity of the formulation. So for that, we have followed this protocol, that is OECD 203 guiding procedure was followed to attain the lethal concentration. For that, we have used Danio Reriro zebrafish. This InstaAMB, aggregated AMB and liposomal marketed formulation we have used, and the multiple from the lower concentration to higher concentration. This fish was incubated in that formulation and mortality was recorded up to 96 hours. So you can see here the result, a 10-fold higher LD50 of InstaAMB was observed compared to the aggregated AMB, which confirmed the enhanced safety. Now coming to the hemolysis studies, that this formulation is intravenous formulation. You can see the InstaAMB and the safer formulation liposomal AMB shows the less than 15% hemolysis, while in case of aggregated AMB, which is a toxic formulation, at higher concentration shows more than 95% hemolysis. So the low hemolysis of InstaAMB propose the safety. So coming toward the efficacy study, we have used some the luciferase transfected leishmania, Donawani promastigods were incubated with the macrophages that converted into the amistigods. After that, InstaAMB and miltefosine is a standard drug for the anti-leishmaniasis, incubated for a 72 hour and luminescence was observed. You can see the efficacy study. Very low IC50 was observed. Low IC50 means the high efficacy. So the 11.22-fold lower IC50 was observed compared to the standard miltefosine drug. In case of cell toxicity is an indicator of safety, you can see 3-fold lower CC50 value. So the 3.66-fold higher selectivity index compared to standard confirmed superior efficacy coupled with the safety of InstaAMB. So the overall conclusion from the presentation, so we have demonstrated in vitro dissolution testing as a practical and useful tool to predict AMB nanoformulation safety. So I would like to conclude the presentation by saying InstaAMB ensuring the safety through monomeric AMB release. Thank you. So I would like to acknowledge my guide, PhD research supervisor, Professor Padmavi Devarajan, and this study was guided under the dissolution study was guided under the Suhas sir, and I'm very thankful to the SOTAX for providing me the facility for USP2 and USP4 apparatus. Thank you so much.
Your words of wisdom have been both enlightening and motivating for all of us. Thank you, Dr. Pooja Torke, ma'am. We truly appreciate you sharing your research journey and knowledge with us. It's inspiring to see young scientists pushing the boundaries of pharmaceutical technology at global level. Now I request Professor Dr. Suvarna Katti, ma'am, IQAC coordinator of Mahatma Gandhi Vidya Mandir Pharmacy College, to please felicitate Dr. Pooja Torke, ma'am. Give a big round of applause. Thank you, ma'am. It's now time for a session with a true of a pharmaceutical quality and regulatory arena. We are privileged to welcome Dr. Rajiv Desai, sir, the visionary leader and expert whose career has been dedicated to advancing pharmaceutical science and regulatory excellence. I request Laiba Sayed to welcome the speaker.
It is an honor to introduce Dr. Rajiv Desai sir, a distinguished leader with over 38 years of experience in the pharmaceutical industry. He has held senior roles in leading organizations such as Lupin, Dr. Reddy's Laboratories, Mylan, Alembic, and many more, and has successfully handled 150 plus global regulatory inspections, including US FDA, MHRA, EMA, and WHO. Currently, Dr. Desai sir serves as an independent consultant and senior technical advisor in quality and regulatory department at the Indian Pharmaceutical Alliance and also chairs the USP South Asia Regional Chapter while serving on the USP Council of Convection. We are truly privileged to welcome such an accomplished professional today. Please join me in welcoming Dr. Rajiv Desai sir. I now request our honorable guest to kindly commence the session and enlighten our delegates.
Backup चालत नाही.